Welcome again on board. I am Francesco Marabita. You know me. I work in the computational medicine unit in Stockholm, but I'm from Italy. And today we're going to talk together about DNA methylation analysis. I tried to prepare some general slides about the meaning, the biological meaning of DNA methylation, and then we will see how can we analyze DNA methylation data. And finally, today we will have a practical hands-on session where you're going to analyze by yourself a subset of the data that have been used for the STATegra project. Okay, so this is the schedule for the presentation. We're going to have the introduction of epigenetics, and then see how do DNA methylation, then small overview of the pipelines, and then the hands-on. So, the classical definition of epigenetics. Classically, epigenetics is the study of mitotically or meiotically changes that are heritable. So, these heritable changes may influence gene function in a way that cannot be explained by DNA sequence. That's the classical textbook definition. But nowadays, we have a more general view of epigenomics. So nowadays we say that the epigenome is the comprehensive view of sequence-independent processes that modulate gene expression pattern. Okay? And we may have different biological phenomena like DNA methylation, histone modification, nucleosome positioning, chromatin accessibility, and chromatin conformation, some of them. All those phenomena can influence gene expression in a way that is independent of the sequence. So sometimes people do this analogy. So if you imagine almost every cell in a tissue, almost every cell in the human body has the same DNA sequence. But on the other hand, cells from different tissues express the same in a different way. And the analogy is this one. So musicians performing a symphony, they use the same score, but different instruments can play a different sound. And that's the analogy. So even though the DNA is the same, even though the DNA sequence is almost the same in every tissue, just as the orchestra perform the same score in different ways, so the cells can express DNA in different ways, according to epigenomic modifications, for example. So sometimes the epigenome is called the conductor of the orchestra, because it's able to give direction for each genes to be expressed or not. And one major form of epigenetic modification is DNA methylation. DNA methylation is the methylation of cytosine in CpG dinucleotides. We also have known CpG methylation, but mainly we have studied CpG methylation. Another interesting form of epigenetic modification is the hydroxymethylation of methyl cytosine. So, and this is a simplified view of the process. Basically, if we start from here, the DNA overall is 70–80% on a mammalian genome. The DNA is methylated on average. So this is usually, but not always, linked to gene silencing. During DNA replication, because you use one strand of the DNA is hemimethylated because the newly synthesized strand that you cannot see here, but it's here, is non-methylated because it's newly synthesized. So there is a maintenance methylation enzyme, methylase, and maintenance enzymes that is actually using the hemimethylated DNA to restore methylation. And this cycle continues through cell divisions. But during embryogenesis, this tissue-specific DNA methylation is reset, so that from the sperm cell, then every tissue a specific DNA methylation pattern. Other key players in the scene are CpG islands. So we have just learned that the genome is 70–80% methylated on a mammalian genome. So if you could look at the profile of the methylation, even by eye, you can identify islands, so stretches of sequence where the DNA methylation is suddenly reduced. Those correspond to CpG islands. They're termed islands because in the genome, in the methylated genome, they're like islands of demethylated DNA. They are approximately 1 kb in length, and they usually overlap with the promoters, and specifically with promoters of constitutively expressed genes, so housekeeping genes. However, genes that are expressed in a tissue-restricted way usually don't have CpG islands. There are different criteria to define a CpG islands. Of course, it is not entirely black and white, but there are criteria to define a CpG island. The most widely used are these, so the length should be higher than 500 base pair, and the observed versus expressed CpG ratio should be more than 65%, and the G plus C composition should be higher than 55%. Given those criteria, we can identify areas in the genome where DNA methylation is reduced. So, because we are interested in analyzing data, so this is a data analysis course, we have to understand what are the axes of DNA methylation variability. Because we don't care about something that is the same across condition, across tissue, across time. So we are interested in analyzing differences. So classically, people have focused on disease. So cancer cell versus normal cells. Or imprinting. I will explain what the imprinting is. But there are other directions. For example, we may be interested in analyzing DNA methylation over time over space. I will give you some examples. Or during developmental processes. And another interesting aspect is how is DNA methylation influenced by environmental stimuli? That is an ongoing field of research. Example of space-dependent variability. By space, I mean tissue or cell type space or genome space. So, sorry for the pictures, but if you have access to Dropbox, this presentation is already online. So if you cannot see it here, you will see it in the Dropbox. Lectures, DNA methylation, and then there's a PDF with the slides. So, this is a principal component representation from this paper, from the data from this paper, and different cell lines have different DNA methylation profiles, or different tissues, sorry. But also, in terms of the genome space, DNA methylation may vary across different genomic elements. This is the variability, and this is the category. For example, we take CpG islands. Their variability is very low because, as I told you before, CpG island usually demethylated. Let's take another example. Enhancers, another category of genomic element. The variability is high. Why? Because different tissues have different enhancers, so different methylation patterns for different tissue-specific enhancers. And then promoter methylation, yes, different variability according to CpG density. So interesting read, take a note. An example of time-dependent variability. CpG methylation may evolve during the lifetime. And this is a brief example, we don't have time, but basically it is possible to build a predictor to predict the age of a tissue based on a bunch of CpG sites. Using about 300 CpG sites, it's possible to predict the age of a tissue with accuracy of 3.6 years. We don't know exactly the reason why, but there is a shift over time for those CpG sites. Other axes of variability, genotype-dependent variability. In terms of DNA methylation, this was, I think, the first integrated approach when they proposed a method for selecting DMPs, differentially methylated sites, that can mediate the genetic risk in rheumatoid arthritis. This is the reference. So imagine you have cases and control, and the methylation is different between cases and control. But you also observe that DNA methylation is genotype dependent. Okay? So, because you know that there is an association between the genotype and the disease, then they use a previously previously available test to screen for DNA for sites that can mediate the genetic risk. So another layer of complexity. Finally, we can say that if we imagine if we imagine biological relationship as networks, basically, we can say that classically we have gene-gene interaction and gene-environment interaction, and the epigenetic regulation is just an extra layer of complexity. Classically, you may have protein-protein interaction or transcriptional networks that can mediate gene-to-gene interaction, or metabolic networks that can mediate gene-environment interaction. But gene-environment interaction, for example, may be mediated by layers of epigenetic regulation. So this is another level of complexity in the data. It's interesting, but I will be very brief on this. If you have questions, please ask me, because otherwise we don't have time. As I was saying, DNA methylation is established during embryogenesis, so that after fertilization, the DNA methylation status of each tissue is fixed during development, so that the brain will end up having a specific brain cells, for example, will end up having specific DNA methylation mark for neurons, and instead prostate cells will have a different methylation profile because they're different cell types. This is not true for imprinted regions. So basically, for the imprinted regions, the DNA methylation is fixed according to the gender of your parents. It's another level of complexity, and we don't have time to go into all details, but for example, there are diseases that are phenotypically different. Prader-Willi syndrome, Angelman syndrome, they are phenotypically different, but they are caused by the same locus, depending the copy that is missing, the paternal or the maternal copy, the phenotype may be different, and this is due to differences in methylation that are imprinted, so specific to the parent of origin, so mother or father. Classical example is methylation and cancer. This is probably the most studied example. DNA methylation is definitely linked to cancer transformation. And you may have both hypermethylation and global hypermethylation. They are supposed to trigger different mechanisms. For example, hypermethylation results usually in the block of the gene transcription of many genes. Instead, global hypomethylation results in genomic instability because many genes that are silenced, or many, for example, transposons that are silenced get activated, and this leads to genomic instability. Another classical example, methylation may be altered by the environment. And there is an animal model to study this, the agouti mice, for example. The agouti mice is a mouse that is able to give different offsprings with different coat color according to the diet. And it was studied. This is the reference where you can find all the details, that this is due to the methylation of a genomic element that is upstream of a gene that controls the color of the coat color. And this is stable. Once the mother is exposed to agents that are able to modify the DNA methylation, then the phenotype will be preserved over generations. Again, later on you can ask me more details, but I want to move forward and go to the data analysis part. Finally, DNA methylation can be modified by the environment, and there are examples of this. So something like a physiological stimulus, like a physical training, physical exercise, is able to modify promoter methylation or enhancer methylation, so methylation a genomic element. And this is nothing as there's no disease involved. This is just the effect of a physiological trigger. Now we go into the data analysis project, but before, it's important to understand some terminology. So sometimes I will talk about DNA methylation, differentially methylated positions, or methylation variable position. Those are sites where, so single nucleotides, where single cytosines, where the DNA methylation is changing, okay? We may also have DNA differentially methylated regions that are regions in the genome where the methylation is changing. Or, as I mentioned before, we may have allele-specific methylation or genotype-specific methylation. Okay, or we may simply analyze regions where we observe variability. So there is no association with the disease, but the methylation is variable, and they may also be interested because something is happening at those regions. Why the methylation is variable at those regions and stable at other regions? Sorry? On average, yes. I mean, the genome is methylated, and CPG islands are not unmethylated. But a region everywhere in the genome may be differentially methylated. So if normally methylated, then it can become suddenly unmethylated during cancer, or CPG island may become methylated. So in this sense, we're analyzing the difference in methylation, not the absolute level. So we are interested in understanding the methylation changes. There can be both hypermethylation and hypomethylation. How do we measure DNA methylation? Different ways. The main task is to understand the difference between a cytosine and a 5-methyl cytosine. The difference is just this one. There's a methyl group added here at this position, at the 5' position, at the 5 position in the cytosine. So we have 3 major classes. We may have enrichment-based methods. We use a protein or something with affinity for methylated cytosine to pull down the regions that are methylated, and then we compare with something with some kind of some other genomic reference. We may use enzymes that cuts at site an enzyme that cuts at sites where we have methylated cytosine, but it is sensitive to methylation, so it doesn't cut if the site is methylated, and then we compare. Or most popularly, we may use the bisulfite treatment. I will explain later on, but after this treatment, we're able to distinguish if a site is methylated or not, and we can use sequencing or arrays as a downstream assay for testing if actually this site is methylated or not. This is basically what I've just said. We'll skip it. There are many factors that affect the choice of the platform. One is the resolution, for example, and of course sequences as 1 base pair resolution. The technique like MEDIP-seq, that is a kind of immunoprecipitation, have resolution in the order of 100 base pair, and the price. This slide is from 2013. So this is a reference from 2013. So they say, okay, it's $10,000 per sample for whole genome bisulfite sequencing. Now this is going down, of course. In 2 years, probably we will see that the $1,000 genome is reality all over the world, and so this is going down. So it is actually a possibility now, but just in selected facilities. And this is a similar slide showing the evolution of the technique. It's there for your own reference. But just remember that also the sites that we catch using different technologies are different, because, for example, arrays, that's answering your question, are usually promoter biased. If this is the whole genome representation, then you can see that the red area is actually increased here. But then it's a trade-off. If we have thousands of samples, we cannot afford sequencing thousands of samples. We choose the array. We have a selected that sample set with 50 samples. Maybe we can afford sequencing. Bisulfite treatment. So it's basically a chemical treatment that allows you to differentiate between methylated cytosine and unmethylated cytosine. Why? Because after treatment by sulfite, unmethylated cytosines are converted to thymines or uracil before PCR. Instead, methylated cytosines are protected and they remain C, even after the bisulfite treatment. Okay. But it's important to remember that after bisulfite treatment, if you start from a homogeneous population of DNA sequences, you may have different DNA sequence if the PCR amplification started from the top strand or the bottom strand. Why? Because what bisulfite is sodium bisulfite is converting is cytosines. So this cytosine is converted to a thymine. Okay, but this cytosine, for example, is converted also to a thymine, but in the other strand you end up having an A. But here, for example, you have a G. Why is that? Because if you use the bottom strand for amplification, in the bottom strand we had a G. Okay? Just remember this at the moment, because it will be a small complication for the alignment. Because if you want to analyze this to the reference genome, your aligner must be aware of the strandness. Because you have actually 4 possibilities, not 2. Experimental protocols. Whole genome bisulfite sequencing. So you start from genomic DNA, then we fragment DNA randomly, and then we do something that's called end repair and after ligation, because during the fragmentation the ends of the DNA may be damaged, so we repair them, we ligate some adapters that are useful and essential in the sequencing process. After possible site selection, when we treat it bisulfite, so we convert the original, the orange DNA in the blue DNA that is shown, light blue DNA, we just amplify at the end in the sequence. The difference with a reduced representation bisulfite sequence is that at the beginning we don't random fragment. If we random fragment the DNA, we don't know exactly where we cut, basically. So we may cut in a region where there is no CPG, in a CPG poor area. So your read in the end will not contain any CPG methylation, maybe, because simply there's no CPG denucleotide. But if you use reduced representation bisulfite sequencing, we simply enrich for areas with increased CPG content, because we use an enzyme, MSP1, that is able to cut at CPG site. The recognition sequence of MSP1 contain a CPG site. So basically we are enriching for areas where CPG content is high, like CPG islands. So reduced representation by sulfide sequencing is slightly biased towards, no, it is definitely biased towards CpG islands. Then the downstream part is it's the same. We ligate the adapters, treat with bisulfite, and amplify with and sequence. There's a modification of this protocol of the whole genome. Classically, this pipeline here requires a lot of DNA. It hasn't been possible. It wasn't possible sometimes to use limited amount of material. But there is a modification of this protocol. So basically, as you can see here, they treat at the beginning with bisulfite. So they create single-stranded DNA that can be tagged and then sequenced. The difference is that here bisulfite treat at the end, here you treat at the beginning. Why I'm doing all this part on this DNA on the protocol? Because the different experimental protocol will affect the quality, the data, and sometimes the quality of the data. And therefore, some specific steps like adapter removal or trimming has to be adapted to the specific experimental protocol. For example, in this case, we already know that we cut with an enzyme. The enzyme basically will cut in a sticky way. That means that there is an overhang. So the DNA will look like this. So before amplifying, we have to fill the gap on the other strand. Therefore, we are actually artificially filling the gaps with unmethylated bases. So when we estimate methylation in reduced representation by sulfide sequence, we may want to remove the methylation data coming from this part, because the methylation is actually influenced by the type of the base that I've used. If I've used unmethylated C to fill the gaps, those cytosine will be unmethylated because I've used unmethylated C. So that's really important in that analysis process. We have to understand what type of protocol we have used. Methylation values. Classically, this is borrowed from the array naming conventions, but we can also use this unfortunate name for sequencing. Why is this unfortunate? I will explain. So there's something called level, and this is simply the percentage of methylation, okay, for each site or for each region. So this is M divided by U plus M. M is for example, is the intensity for the methylated intensity, or the number of reads supporting methylation. U plus M is the total read number, so the reads supporting unmethylation, the reads supporting methylation, or it may simply be the total signal intensities in array data. Sometimes this quantity is not convenient to use in downstream analysis, so a popular transformation is the log-it transformation of the beta value that is called N value, borrowed from the array M values, basically. And there is an interesting property, so that the beta values has non-constant variability. The variance, the standard deviation in this case, is influenced by the average. There was some blue here that you cannot appreciate, but because of the projector. But the value, the value of the standard deviation is influenced by the mean value of the beta. This is not the case for M values, and this is an interesting property to use, for example, linear models. Let's talk about analysis pipelines. Like any analysis pipeline, we can identify steps, and for each step, we have recommendations, we have different views, and we have different approaches. This is a review from 2012 where all the steps are analyzed, and for each step you will find interesting references and links to website and tools that you can use. It's general for DNA methylation data, not restricted to sequencing or arrays. So first of all, we measure, and we measure using the assay that we choose. So microarrays of sequencing or enriched base methods, the three arrows on my previous slide. Then we analyze, pre-process, normalize, and detect differential methylation. Then we visualize the data. Data visualization is also an important tool of the step of the analysis pipeline. Finally, we can validate and interpret. And by validation, I mean technical and biological validation. Okay. This is a typical analysis pipeline that will take you from FASTQ files to reporting differences in methylation. Now you should be familiar with FASTQ file. We have seen this morning what a FASTQ file is and what a quality score is. We will go through quality control as we have done for the RNA sequence data. Alignment, same as we've done with this sequencing data. Once we have BAM files or some files, we have to do an additional step. So the BAM file will specify the location of the reads on the genome. But we have to extract data from the BAM files. And we can do this because we have converted the reads we have converted DNA with bisulfite. So the reads will contain the information whether or not the site is methylated. But we have to use special allelers that are aware of this modification. Then we have TXT files, something very simple that we can read, where I have an estimate of the methylation and possibly the number of total reads that support that site. Then we go again to quality control. As David was mentioning, every step you do, it's always good to perform a quality control step that is specific for that step. We may or may not perform smoothing. I will explain later. Then we evaluate methylation across genomic position. And finally, we proceed to differential methylation analysis and functional analysis, and we report the finding. The quality control. You already know what is the goal of quality control. So to trim low qualities and adapter contamination. If you think about the bisulfite conversion, I have a slide. You may notice that somehow the complexity might be reduced because some of the original Cs are actually converted to Ts after bisulfite treatment. But in the reference genome, you don't have a T, you have a C. Okay? So this is something important to think about and it's important for both quality control and alignment step. Because we have to be we want clean data. By default, the alignment would be lower. Why? Because you have a reduced complexity in the reads. Sometimes, instead of using 4 bases, use 3 bases because we have converted one C to T in the reads. So the alignment rates are lower. Therefore, we want to be extremely clean. So the quality control step here, it's important. So we eliminate the cycles where the quality is lower than a certain threshold. For example, here I've used 28 as a threshold because it's important to remove low quality bases because the alignment rate is it's lower by default by using reduced representation bisulfite. Then there are some protocol-specific actions. I already explained this. So in reduced representation bisulfite sequencing, we start by digesting the reads, the genomic DNA with a restriction enzyme. That is cutting like this. When we fill the gap, we artificially modify the status of the methylation in the ends. So we will never use, it's recommended not to use the end of the reads to estimate the methylation sequence, methylation estimates. So We may also trim for this part, depending on the read orientation. This is the effect of the adapter contamination. If you have an adapter, the alignment rate will be lower. You want to get rid of this, and you can detect this effect by looking at the sequence composition. So you have 4 different bases. By the end of the read, the content of what is the blue, I cannot see, but let's assume it's a C. The content of the C is artificially increasing because maybe the adapter has a different composition in terms of Cs than the genomic DNA. So you may detect this, but once you trim, after adapter trimming and quality, then the sequence content is the same across all cycles. And those are data from STATegra, for example. Again, this slide, because I want to stress the concept that we have to use an aligner that is aware of this process. If you use a common aligner, then we end up discarding many reads because they will not map to the genome after bisulfite treatment. Is it clear why? Yes? Okay. So there are 2 strategies, basically. One very popular strategy that includes the Bismark aligner is to use a 3-letter aligner. So we convert all Cs into Ts, both in the reads and in the reference genome. It's a very simple but effective solution. So instead of using the 4 letters, we use 3 letters because we have artificially converted all the Cs into Ts, like an artificial bisulfite treatment. In this case, you have no problem because in principle there should be no mismatch unless you have a SNP. Another choice is to use methylation-aware aligners. Basically, in the reference, you change the Cs with the Y character, that is a wildcard, that will match both Cs and Ts. Or you modify the scoring matrix, so when you calculate an alignment score, you don't penalize the genomic C that is matching with a T, simply. This strategy, the 3-letter alignment, is the strategy Bismark used, and Bismark is a very popular alignment tool that I also use for the STATegra data. This is how it works. After the bisulfite treatment, basically you have the C to T conversion or the G to A conversion in the bottom strand. So because you have actually 4 possibilities, in parallel you align your read with all the 4 possible combinations. So the top strand and the bottom strand. So you align these to this, to this, align this to this, and to this. So for each read, you actually have 4 references, 4 alignment processes, and you later on check which one of these is the best alignment. Therefore, in this case, in this example, the alignment of this read corresponds to the 1, the arrow number 1. Later on, you will end up calling the methylation because you can, if this is the genomic, the genomic reference, unmodified, then you can count the mismatches and say whether the site is methylated or not. Can you see that? Okay. And that is an easy way to count methylation or to quantify methylation. The disadvantage is that because we're using 3 letters, then the complexity of the library is reduced, and therefore the alignment rate is not 90%, but something around 70%. But it works. A lot of quality control. Now let's say we have done the alignment. We're here, somewhere here, we have the BAM file. But still, we're not ready to do differential methylation. We had to go through additional quality control, like we do for RNA sequencing. First, we do this. We check that the DNA methylation is there's no any bias. There's no bias according to the cycle, because in principle the percentage of methylation should not depend on the cycle, on the sequencing cycles. If we do detect something, it's because of artificial biases. Reduced representation bisulfite sequence, first few cycles have no methylation. Then we exclude the cause from the first few cycles. Another important thing is to evaluate the coverage. You perform your experiment, you plan everything homogeneously, but sometimes you may end up having one of the samples with very low coverage. So if you include all the samples together, then one of the samples may be actually a limiting factor, because if you select sites that are covered in all the samples, then you have one limiting factor, and then you end up excluding a lot of sites simply because in that sample you don't have the data. So evaluate the coverage is really important, that you can identify outliers that maybe you want to inspect, or you may get more data for this sample only because you don't have sufficient data. Finally, use the same tools as that's used for RNA sequencing for doing clustering, where you can identify possible outliers like this. Imagine if those are two groups. They cluster nicely according to the group, but this sample is not clustering nicely. So why? Then you investigate the possibilities and eventually include or exclude the sample. Smoothing. It's an additional step that is specific for DNA methylation. DNA methylation has an important property. The methylation level neighboring CpGs is locally correlated. That means if a site is methylated here, is highly methylated here, that means that it's also highly methylated in the neighboring areas, because it's a biological phenomenon. Then you may use neighboring sites with high coverage to basically predict a better methylation for the site with low coverage. Ideally, if you don't do the smoothing, you have to exclude samples with the low coverage. Let's say if you have 4 reads only, the methylation estimate can be wrong, totally wrong. So you have to set a threshold on the coverage. But if you do this, you discard a lot of CpG sites, unless you have sequenced with an average cover of 30x, that it's more expensive. So there are approaches to smooth the data and use more sites and not discard any sites with low coverage. It has been shown that these can improve precision even with low coverage samples. Even if the coverage is around 4x, you may increase the precision. And basically there are 2 main frameworks. One is for whole genome bisulfite sequencing, that is the B-smooth process contained in the BSSEC package, and another one is for targeted bisulfite sequencing or reduced representation bisulfite sequence, and this is called BISEC, and it's the package has the same name. A smoothing function is modeled only within clusters of CpGs. Okay, we'll see this example in the hands-on because we have reduced representation bisulfite sequence, so cluster data. We're not sequencing the whole genome. Finally, we're ready for testing. Ready for testing differential methylation. But I want to remind you that we can test genomic positions, so we can have differentially methylated positions, or more interesting, we can test for regions, because we may expect the methylation is not changing at a single site, but on a bigger region, and in this case we may we end up calling DMRs, differentially methylated regions. Or we can calculate the average methylation for genomic elements. Let's say I have a promoter, I take the average methylation of the promoter or the enhancer. It's another alternative approach, depending what's the analysis. Yes. Okay, I have 10 more minutes basically. The goal of differential methylation is obtain a list of significant DMPs or DMR, and there are different methods. Originally, people were simply doing a Fisher test. So if you have your count data, the reads supporting methylation and the reads supporting non-methylation, you can simply do a contingency table with cases and control and count methylation, non-methylation, 2 by 2 square table, and then you do a Fisher test. This is okay, but there's a main disadvantage that in this case you cannot model any biological variability. You simply have two groups, you average the reads. The biological variability cannot be modeled. So you can use instead logistic regression on transform methylation values, the famous M value that I showed before. Or you can do beta regression. So the DNA methylation profile has a beta distribution. That's why we call beta the methylation level. So instead of using linear regression, you can perform beta regression. This is a more advanced approach. Or another interesting approach that's been used a lot is the bump hunting approach. So because genomic methylation values are correlated across the genome, you may look for regions in the genome where you observe an effect that is going in the same direction. So if the blue samples are the normal samples, and the other sample, the red, should be red, is the cancer sample. So there is coherent change in methylation, and for example in this case it's the mean difference. If you plot the mean difference across the genome, then you will see that there is a bump. So the T statistics or the mean difference in methylation bumps at this position according to the 0 level, let's say. So you set a threshold and then you call these differentially methylated regions. And they suggest also a permutation approach to calculate an FDR for this. But it is the permutation approach is complicated by the fact that sometimes you have additional covariates, so it's not always possible. Finally, you perform functional analysis and reporting, and there is a list of tools that you can use. This is still from the Nature Genetic Reviews, and I will show you one example of this, the GREAT analysis. That is a very popular tool nowadays. I actually will start from here. Classically, when we have genes, we have usually no problems in doing the functional analysis, the gene ontology enrichment, because function is given by genes. The gene is associated to a certain function. But we have a problem when we have differentially methylated region or SNPs or ChIP-seq regions. So the function is driven by the gene, but how can we associate our genomic region to the gene? There are very simple rules. You can usually take the closest gene. That's an option. But maybe you have complex genomics environment where that genomic region is not only linked to the closest gene, but also to a distant gene. So the approach in GREAT is to use gene regulatory domains. Each gene is assigned to a basal regulatory domain according to this transcriptional start site. You have certain rules, and you can define this tick area here, an important regulatory domain for the gene. Okay? Then you extend this region upstream and downstream until you reach another basal regulatory domain, so that one region, for example in the genome, may be actually associated to multiple genes. Okay, so the blue triangles are actually the ChIP-seq regions or the DMRs. And instead of doing a hypergeometric test, you can do a binomial test taking into account how many regions you have, what is the fraction of the genome annotated with that ontology pi, and what how many genomic regions are annotated with the ontology pi. So this is also a method for correcting for the fraction of the genome that is associated with a certain ontology. So as a result, one genomic region, one DMR, may be associated with multiple genes, and that is possibly a better way for doing the gene ontology analysis with DNA methylation data. Lastly, the batch effect. So this is the publication that David was mentioning this morning about batch effect. DNA methylation has many high throughput data is affected, sometimes very highly affected by batch effect. A batch is a subgroup of experiments that are associated with a known biological sources of variability. Let's say different lab technicians, different laboratories, different library preparation days. Those are batch variables. They can influence variability in those data, but they're not correlated with the biological variability. So, and this is the famous study where basically analyzed normal individuals that were perfectly clustering into subgroups, and this is very common. You may see this type of pattern many times when you analyze the high throughput data. Then, even though normalization has been performed, and overall you will say quantile normalization is performing very well, still there will be some genes that behave differently, and those genes are not corrected by global approaches like quantile normalization. They will drive this clustering. But the advantage is that from high throughput data we can estimate this effect, because we're measuring any gene, so we can actually model the batch effect and reduce the batch effect. Classical representation, you have, this is from array data, not sequencing data, but it's to convey, let's say. No batch adjustment. You have males and females, that's given by the color, and you have batches. So B and A. You can see that without batch effect correction, the main cluster, there is the first component is actually correlated with the batch A and B. But when you correct, for example, for a batch, the female sample, or the male sample that was in the batch B is actually going with the correct batch, with correct biological variable, in this case the gender. Classical example. So it's important to correct for batch effect also in methylation data if you have any batch effect. But a huge recommendation is do the best experimental design you can do, because if you design your sequencing experiment, you have all cases in one library preparation and all controls in the other library preparation, then there's no batch effect correction, because the batch is simply confounded with the biological variability. That's all, and thank you for listening.